Thank you very much for the introduction and also for the invite to present today. Shall I share slides from my screen? Yep, I'm taking that as a yes. All right, wonderful. Many thanks. Okay, so yes, as it was said in the introduction, I will be today presenting work that we've recently published using our human liver chip and how we've been able to use it to predict drug-induced liver injury. And then importantly, what we would really love to see with organ-on-a-chip technology, of course, is that it's incorporated much more into laboratory workflows. And I'll talk a little bit about that. And given that we're in a 3 hours symposium, I'll show how we believe that the incorporation of liver chip into routine workflows may also start to have an impact on the 3 hours. So very briefly today, I'm just going to touch on, the reasons behind why patient safety requires modernisation or perhaps new tools such as the liver on a chip. And we'll talk about the performance assessment that we conducted with our liver chip model, how it implements into workflows, and then a little word on the end about model variability and reproducibility, which is quite important for encouraging people to think about incorporating these models into their work streams. And I'll close with a few final remarks. So many of you know that in 1938, in the Congress passed the first US federal drug, Food and Cosmetic Act, and this mandated animal toxicity testing. And of course, this was in light of the sulfanilamide disaster that had happened the previous year. And it really brought forth the need for developers of drugs, even new formulations, as was the case in the sulfanilamide example, to think about the safety of these new drugs as they move into humans, rather than just, of course, focus on efficacy. And that act has been with us for many years, and animals have definitely performed a really important role in helping move forward many of the therapies that we see on the marketplace today. However, it's become very apparent that these animal models, whilst they do a good job in many cases, there is still room for improvement. And this slide outlines a few of the more recent examples, analysing the concordance between the animal studies and human outcome. This has then culminated in a modernisation of this act. This was signed into law on December 29th last year by President Biden. And this now encourages users in the pharmaceutical industry to consider non-animal alternatives as they progress their candidate submission through the IND process. So this is quite a landmark, actually, for people like myself working in the organ-on-a-chip field. So why is predicting toxicology so challenging? Well, we've got a couple of main reasons that we like to talk about. First and foremostly, toxicity is not only species, but also organ-specific. And so we've shown, and again, we cite some more literature here, and that animal models are not fully, equipped, if you will, to always be able to predict the toxicity. Also, drugs, especially drugs that we're developing today, often have pleiotropic effects, but moreover, drugs are also increasingly used in combinations. And actually, there's been some important work done of late showing that drugs, antivirals, particularly for the treatment of hepatitis B, when assessed in isolation, they're completely safe and they have efficacy. They have better efficacy, though, when these two antivirals are combined. However, the combination of these two antivirals actually has led to patient fatalities due to liver toxicity, which is really the purpose of today's presentation. There are predictive gaps then, partly driven by the species differences. These gaps exist in the in vitro models. conventional in vitro models, that's where we grow cells in static culture, usually in 2 dimensions. These also don't recapitulate the organ level complexity. And AI is all around us nowadays, and certainly from an in silico perspective, the predictions are improving all the time, but nonetheless, these predictions really still need to be confirmed in some form of biological model before fully progressing an asset into the clinic. So in terms of drug development, then what's the cost of the status quo? So if we just keep doing what we're doing, we'll still see similar figures as listed here in this slide. So about 75% of today's R&D costs are due to the cost of failure. 40% of clinical trials are failing due to toxicity, and around about 50% of post-approval market withdrawals are also due to toxicity. So we need to find better ways to tackle this. At Emulate, we've been focused particularly on the liver, and we follow the reports, as many of you may do, of companies who have to declare that a clinical trial has been paused, or there have been issues found in a clinical trial. And here's an example from March last year with Aligos, where actually they had to stop their hepatitis B med after an unexpected reaction in 4 patients. Now, not only does this have a patient safety concern, it also caused 80% of the company value to be wiped out, and up to this point it was estimated that the company had probably spent around $50 million on the clinical process. So if nothing else, this aspect also needs to be addressed. And now here on this slide, you'll also see some other statistics that talk to not only clinical trial failures on the left, the pie chart in the middle shows post-market withdrawals. And here highlighted in blue, you can see that the hepatic causes of clinical trial failures make up about 13% overall, and about 21% of post-market withdrawals. So this is a leading cause of late-stage drug attrition. And on the right-hand side, a rather depressing look at some drugs that, albeit were approved in the late 1990s, early 2000s for different therapeutic indications, have actually gone on to cause patient deaths and or liver transplants. And then lastly, just to really bring the point home, as I mentioned earlier, we continue to monitor the drug-induced liver injury and see this as a patient safety concern. And this example here shows you that even as much as in the last 12 to 14 months, there have been several high-profile examples of where clinical trials have been stopped, and indeed two patients have died as a consequence of current testing approaches towards assuring safe patient safety. So hopefully I've convinced you then that there really is a need to have some better models to help us get better candidates into the clinic, and of course protect the patients involved. So this was really part of the inspiration for looking at our liver chip model. What we have been doing over the last 18 months or so at Emulate is using guidelines actually that were published by the IQ-MPS consortia. So for those who are not familiar, the IQ is it's a group of, it stands for Innovation and Quality. It's a pre-competitive group that pharma companies can join, and their goal really is to drive innovation and qualification. And they want to do that in a very pre-competitive or non-competitive manner, such that new technologies can reach the companies and they can be utilised to improve the drug discovery and development process. We've published this work in Nature Communications just at the end of last year. That's the reference there. And over the next few slides, I'm really going to bring this paper to life for you. So first of all, our chip that we build is what we call a quad culture chip. It involves 4 key cell types that you would find in a liver sinusoid. I'm going to show you a little cartoon of our chip in just a minute, but quite simply how we set this model up is that we have our hepatocytes situated on the top channel of our chip. These hepatocytes are sandwiched in between 2 layers of extracellular matrix, and they have a porous membrane that separates the top channel from the bottom channel. So this culture arrangement, also known as a sandwich culture, is very typical of hepatocyte culture. It enables the hepatocytes to correctly polarize, and indeed you can see the bile canaliculi formation as well as these cells really become embedded into the chip. In the bottom channel, then we seed 3 additional cell types. We seed the stellate cell, the liver sinusoidal endothelial cell, and the Kupffer cell. And we're trying to be honest to that spatial anatomy. So we have the stellate cell sitting above the endothelial cell, so using really the porous membrane to represent the space of Dease. We have the endothelial cells and then the Kupffer cells below, which of course are the macrocytic cell in the liver. Throughout, we use primary human cells, and indeed over the course of our investigation I'm about to present to you, we've used 3 hepatocyte donors. So I'm just going to show you a quick image of a video of our chip, just really to bring this to life for people who are not as familiar. So as I mentioned a minute ago, our chip has 2 fluidic channels. The top channel is 1 millimeter by 1 millimeter. The bottom channel here is 1 millimeter by 0.2 millimeters. As I mentioned before as well, these channels are separated by a porous membrane. The pores here are 7 microns in diameter. This membrane is then coated with an extracellular matrix specific to the cells that we want to seed. So typically we put an endothelial cell on top and an endothelial cell on the bottom. Unique to our platform is the ability to apply a mechanical stretch, which you saw there with the vacuum channels. The 2 main channels where we seed the cells are fluidically independent. This means that we can flow optimized media for the cells present in that channel. We can also remove the media from the top channel of the chip to create an air-liquid interface if that were needed for models such as the airway, for example. So as I mentioned, we used guidelines from the IQ-MPS consortia. They set about producing a series of manuscripts which guided folks like myself to build an appropriate liver model. When you look at this particular paper, it's essentially broken down into 3 stages. First of all, then they're asking developers really to take a look at baseline functionality of the chip. So for example, albumin production, urea synthesis, and gene expression. Then one would move to a more in-depth analysis around biomarker stability, enzyme activity, and tissue histology. And then lastly, and importantly, they proposed a set of 20 drugs to test in the model. That these drugs were all drugs that had been in the clinic and therefore were known to be either clinical DILI positive or clinical DILI negative. So this was the study really proposed by Bodie et al. In 2020, and it very much focuses on a qualitative assessment of a liver chip model. However, we also wanted to perform a quantitative assessment because we want to provide guidance on how these chip models could be used by pharmaceutical scientists in their everyday workflows. So what we did was we added to the test set proposed by the IQ-MPS with some further small molecule drugs that had also been in the clinic. But importantly, these small molecule drugs had also been tested on 3D hepatic spheroids, which is another in vitro model that many scientists are using to do some early safety screening in a project's lifetime. What we found was with these hepatic spheroids is that they actually have a high false negative rate. So we actually were able to enrich our drug test set with these drugs that were deemed false negatives in the hepatic spheroids, with the aim really of understanding how was the performance of our model versus other cellular models that are used today. You can see then that of the 27 small molecule drugs that we tested in the table here on the left-hand side, that 22 of them are clinical DILI positives, 5 are clinical DILI negatives. They also cover the major mechanisms of drug-induced liver injury. And this is important. This is in line with guidance from another consortia, the MIP-DILI consortia, who published, again, similar sort of guidance on how one might address this challenge preclinically. And certainly their guidance and their recommendation was to include any assessment of a model with drugs that represented these major mechanisms. Throughout our study then, all of the drugs were tested blind by our scientists, and the concentrations that we tested in were adjusted for protein in the chip media. And lastly, the way that we set up our concentration range is we obtained the human free Cmax from the literature, and this is the human Cmax that was deemed to be causing efficacy for that particular drug. And we said, okay, we'd like to test that concentration and then go to a concentration up to 300 times that value. Now, in most cases that was possible. In a couple, it was not. And the reason it was not was due to solubility challenges. So what did our experiment look like? Well, first of all, we used 870 human liver chips across 5 experimental cycles using 3 different hepatocyte donors. We have a period of around about 5 days where we activate our chip. We then coat and seed the cells, starting with the hepatocytes, and 24 hours later, we seed in the stellate cells, the sinusoidal endothelial cells, and the Kupffer cells. And at that point, 24 hours later, we attach our chip into our fluidic platform and begin the flow of media. At that point, we call that day 0. And on day 0, we are also going to be adding the vehicle controls as well as any compound that we want to test. And the way that our platform works is that it's a unidirectional flow of media. That means that the media containing the vehicle or the drug flows through the chip and is collected in a media reservoir on the outlet of the chip. This can be collected, and we did collect the media on days 1, 3, and 7, and we were able to use that data to measure albumin in the media, ALT in the media, and urea. Throughout, we also continued to look at morphology of the cells using bright field microscopy. At the end of the experiment on day 7, we then took some terminal endpoints to look at caspase 3,7 as a marker of apoptosis. We looked at mitochondrial integrity using the stain TMRM. We also stained for Dipro-Red to see any accumulation of lipids in the hepatocytes, as well as MRP2, which is a drug transporter, and that just really shows that we have polarized hepatocytes. So this is our testing arrangement. We lastly did some RNA-seq, and we performed this on days 3 and 7 and compared those to the freshly thawed hepatocyte donor. Okay, so let's look at the data then. So if you recall back to the IQ-MPS consortia, they first of all asked for the demonstration to show typical hepatocyte phenotypic markers. So we look on the bright field microscope. First of all, hepatocytes are usually cuboidal, sort of hexagonal type cells, nice plump nuclei, which you can see here. So very happy, very healthy. The non-parenchymal cells are in the lower channel, and those uniform circles you can see are actually the pores of the membrane, and you can see polygonal shaped cells below. It's a little bit difficult under bright field microscopy to really pick out the different cells, and that's really why we use immunofluorescent stains, antibodies, and use confocal microscopy to identify those. So those images on the right-hand side then, on the upper channel, you can see we've stained for the cytoskeleton in green to start to see some of that branching bile canaliculi structure, and also the MRP2 next to it, which would be present in that region. You can see that then the hepatocytes have a lot of mitochondria. This is important because they do need a lot of energy. And then down below, the endothelial cells were stained with CD31, the stellate cells are stained with vimentin, and the Kupffer cells are stained with CD68. We also took one step further and looked a little bit deeper at these cells using transmission electron microscopy. Here you can see on the left-hand side, actually 2 hepatocytes next to each other. You can see the thin bile canaliculi structure. It's about 1 to 2 microns in diameter. And you can see lots of dark spots here. These are deposits of albumin. On the right-hand side, you can see the endothelial cell with a Kupffer cell sitting below it. The Kupffer cell is obvious due to lots of its RER as well as pseudopodia on the right-hand side, whereas the endothelial cell has got lots of lysosomes again, which is very typical of this cell for its endocytic capacity. Next, we had to demonstrate that our liver chip would produce the baseline markers. Albumin is routinely used as a marker of hepatocyte health and baseline functionality, as is urea. And the dotted lines on this particular slide really indicate some levels that have been computed. So these are estimated levels of what you would expect to see in an in vitro system and where, and how that might translate to in vivo. So in other words, what we're looking for is albumin and urea production to sit in between these 2 dotted lines. The individual dots on each bar are representative of individual chips, and you can see donor 1, 2, and 3 colored differently. So across the 7-day period, we can see that albumin is well above the lower level throughout the first sort of up to about day 5, really. And then on donors 1 and 3, it drops slightly below that 20 microgram per day per million hepatocytes. So that kind of tells us that we probably wouldn't want to have these hepatocytes in culture for much longer. By the time we get to day 7 on the chip, we're about day 12 or day 13 in culture. So these hepatocytes are getting on. On the right-hand side, you can see that our urea production, especially in donor 1, exceeds that we would expect to see physiologically. This is a phenomenon we don't fully yet understand, but nonetheless, it does indicate that these hepatocytes are functioning in a healthy way. The IQ also asked us to look at gene expression, and the gene expression here is particularly important because they chose 17 genes that represent major drug metabolizing enzymes as well as drug transporters. And one of the first things that typically happens when hepatocytes are in culture is they switch off a lot of the gene expression for these key drug transporters and drug metabolizing enzymes. And because many of the mechanisms that lead to drug-induced liver injury involve a hepatic enzyme doing some drug metabolism or a drug transporter either being inhibited, most of the time inhibited, I should say, then of course, if the genes are not expressed, the protein won't be translated and therefore the cells won't be as functional. So that was the reason why they were asking us to look at it. And you can see here the data is actually expressed relative to the freshly thawed donor. It's the closest that we could get to how it might be in vivo, and you can see very stable expression in both donors for the majority of these particular genes. There's a couple of examples where maybe the levels actually decline a little bit over the culture period, but nonetheless nothing that we're terribly worried about here, and certainly gives us confidence that we'd be able to pick up drug-induced injury mechanisms due to metabolism. The next thing that IQ wanted to really understand was how well could a model distinguish between 2 drugs, one that was known to be toxic and one maybe lesser toxic or even not toxic at all. In this example here, we're looking at trovafloxacin in the filled circles and levofloxacin in the open circles. And why this is important is because, again, if you're going to use this in the process of discovering new drugs, quite often you'll be looking at drugs with a similar chemistry, and it's important to be able to rank order those in terms of their likelihood or potential liability to cause toxicity. So here you can see that trovafloxacin on the left causes a concentration-dependent reduction in albumin release. It also causes a concomitant increase in ALT release. ALT is a transaminase and it's a clinical marker of hepatic injury. And you can also see that there's a concentration-dependent increase in a morphology score. So at Emulate, we've developed a morphology score that allows us to see how healthy these hepatocytes are. Basically, the lower the number, the healthier you are. The higher the number 4 means that, you know, you're not really in great shape at all. So again, you can see that trovafloxacin increases over time, over concentrations, I should say. Whereas the dotted lines here are representing levofloxacin, and you can see levofloxacin's without effect on albumin, ALT, or morphology. So this clearly shows us that Liverchip was able to distinguish this between this pair. And moreover, when we did one additional step here, which was we used caspase 3,7. It's a marker of apoptosis to really be a proxy for the trovafloxacin toxicity. And you can see here that, again, with increasing concentrations of trovafloxacin, we're able to see a caspase signal, whereas if you use levofloxacin, there's really no indication of caspase staining over those concentrations that were tested. So all that's to say that we were able to correctly pick trovafloxacin toxicity over levofloxacin. Remember, my scientists were all blinded when they performed this piece of work. Now, IQ actually, not only did they propose this one pair, they actually proposed 7 matched pairs, and you can see all 7 pairs here. And this table is just simplified to show you the changes that were measured in those 4 parameters. So you can see all of the more toxic drugs on the left-hand side of the slide here all had a reduction in albumin. All of them, bar thiouridine, caused an increase in ALT and morphology score. And in terms of imaging, we were able to see either apoptosis or mitotoxicity and some steatosis when it comes to thiouridine. So we were very happy with these results. But as I mentioned right at the beginning, that was really the end of the guidance from the IQ. It was very qualitative in focus, and we wanted to perform a quantitative assessment as well to really add some additional value to scientists using the platform in their own workflows. So what we then did was we said, okay, we understand now Liverchip has indicated of these 27 drugs, we're able to say which ones we believe are actually toxic and which ones we believe are non-toxic. So we did a simple sensitivity specificity analysis using the 2x2 table that you see on the left-hand side. And first of all, we started off with animal models. So we don't actually have the animal model data for the 27 drugs that we tested. However, we know that those, because it's a requirement to progress a drug into the clinic, these drugs have all been in the clinic, we know that animal model data must exist. However, we also know that the animal model may have incorrectly classified the safety liability of 22 hepatotoxic drugs. So we set the animal models as having a sensitivity of 0, because if it was not 0, then it would have picked out the hepatotoxic drugs in the clinic. Because we wanted to compare to hepatic spheroids, we were able to also pull data for 22 of our 27 drugs from the literature. That was using 3 different papers, each of which had used different hepatocyte donors and had tested in excess of 100 drugs. And of those 22 drugs, we know that 19 of those drugs were actual clinical positives, 3 were clinical negatives. The hepatic spheroids were correctly able to identify 9 of those. They missed 10 of them. Remember, we did enrich our set for those drugs that were deemed false negative. So this was not unexpected. And this therefore came out with a 47% sensitivity for the spheroids. Now, when we were looking at our Liverchip data, what we discovered actually was that in the first 2 donors that we tested, we had 18 of the 27 drugs in common. And we said, okay, when we use 2 donors in our Liverchip, 15 of those drugs are actually hepatotoxic, 3 are negative. What is our sensitivity? And we found actually that Liverchip was able to correctly identify 13 of the hepatotoxic drugs, and this gave liver chip a sensitivity of 87%. Another measure that we did to understand the performance and the predictive value of this particular model was we performed a rank ordering exercise, and we were able to calculate that liver chip had a Spearman correlation coefficient of 0.78. And this indicates then that it's a highly predictive model. So let's now move to the sort of last section of this presentation, which is, given the performance of the liver chip we've identified, how might one actually think about using this in drug development process, and how, given that we're here today to talk about the 3 Rs, how might it also impact the 3 Rs? So what I've shown you here at this point in time is a very traditional screening cascade. So at this point in the process, we are just about to enter lead optimization. We'll have performed traditional in vitro models on our candidates that we're interested in, and we'll now have 2 or 3 that we really want to progress through an animal dose range finding study into a GLP study, and then onwards into the clinical trial. Now, that's the path that I've taken myself and is still practiced today. And what we know is that 90% of candidates that enter into phase 1 fail to make it to the approval status. So as I was saying at the beginning of the presentation, it's really important for us to find methods and ways to improve that statistic. So this is where we think liver chip would come in then. We've performed our traditional in vitro models. We would then situate liver chip before doing any of the animal dose range finding studies. And the reason for doing that then is that if we find drugs that have an overtly toxic signal, our recommendation would be to the project teams to have a real stop and think about whether it's worthwhile to progress that drug into the clinic. If the team looks at all of the other factors that they would look at during this type of discussion, and they say, actually, yes, we don't think we should progress that, then of course it would not go into an animal study, and we would start to see a reduction in the number of animals used. But of course, it is important to remember that animals will tell us much more about organs other than the liver chip. The liver chip's only going to tell us here about the liver. And we're saying that if we follow such a path, we would be able to enter higher quality candidates into phase 1. And remember, I did show you many examples, and even as recent as this year, where clinical trials have been placed on pause due to hepatotoxicity. So some people sometimes say to me, well, okay, so I can put liver chip in the process as you've described, but, you still are only going to get a toxic or a non-toxic signal. And so this is where we've actually been doing some further thinking, and this is currently unpublished data. We are in the process of writing this up for a follow-on publication. But it really speaks to the fact that liver chip will provide the user quantitative output. So if you remember, what we're doing is from our endpoint analysis, we're measuring albumin IC50, we're measuring any ALT release, and we're looking at the morphology. That, together with the known Cmax of that particular candidate drug, or if you're pre-clinic, you will have a modeled range where you believe you need to be to see clinical efficacy. So we use that concentration. This allows us to calculate what we are now terming the liver chip DILI score. And the nice thing about the liver chip DILI score is that we can map this to industry standard DILI risk categories to provide the user with some additional context on what the liver chip data may lead to. And that's what this slide really shows. So here, again, this is all unpublished data. We've taken the data from the 27 drugs that we tested in our liver chip, and we mapped the liver chip DILI score against the Garside score prediction. So this is a positive predictive value. And essentially what Garside described in her work was that if you have a score of 1, that drug, and this was based on retrospective analysis of nearly 300 drugs actually, if you have a Garside score of 1, you are almost certain to cause death or acute liver failure. And most of these drugs were withdrawn from the marketplace or received a black box warning. And so what we would be saying to users, if you have performed the liver chip and you see a liver chip DILI score of 31 or less, that every time we saw a score of 31 or less, we map to a drug that was labelled a Garside 1. So in that way, that would be a real stop and think for the project in terms of whether they really wanted to progress. And what you can see with the increasing thresholds in terms of our liver chip DILI score, you can see that the score prediction is moving to the right. In other words, the higher the liver chip DILI score, the less likely this particular candidate is carrying a hepatic liability. And then finally, when we get to the threshold of 375, any liver chip that generates a score above that, we're saying that the drug in this case is very unlikely to identify a DILI risk. But don't be alarmed, there is a 40% value shown in the hash box there under the Garside 2 score. And this really represents the no model is perfect, ours included, and this does reflect the 80% sensitivity that, of course, we reported out for the liver chip. So putting that together, then we were saying that we can now give a more quantitative assessment to users to say, depending on that liver chip DILI score, we would either advise you to stop or to evaluate or indeed to proceed. And we hope that those guidance will really help drive adoption and usage. And in the last few slides, I just want to turn then to thinking about what else is important to drive adoption. It's thinking about model variability and reproducibility. And on the left-hand side of this slide, I'm citing 2 papers that have been published by the FDA. It's in the food side of the Food and Drug Administration. And this was a result of the first CRADA that Emulate had with the FDA, which was established in 2017. We've since established a second CRADA. We've extended it to finish in 2024, and it now encompasses CEDAR, CBER, CVM, as well as NCTR. During the publication then of these 2 papers, the team at CIFSAN put our liver chip through a series of performance metrics and looked at the PDMS substrate and its ability to bind drugs, its sensitivity and specificity, sample size, variability and repeatability. And indeed, their conclusion was that actually liver chip does predict toxicity, has a high sensitivity and specificity. And in terms of our sample sizes, we were around 3 to 4, depending on the endpoint that you wanted to measure, although they were a little bit concerned about the PDMS material, which we understand. So this was really nice external endorsement that our platform can be situated in an external laboratory. Scientists can be trained and to run and find similar data as we've just reported out. To back this up, then, we've been following one of our hepatocyte donors over the last 18 months. We've performed 23 different internal studies with this donor. And here the graph is showing you the albumin production on day 1. You can see there that on the whole, this albumin production is fairly stable. However, study number 8, actually something funny went on with study number 8, which we're still trying to understand. But without study number 8, actually, we've got about a 96% reproducibility rate. And we believe that our reproducibility is high because we have very good SOPs in place for cell lot testing and operator training. So as I close out then, one thing I'd like to leave this group with is thinking about how we could start with a roadmap for adoption of next generation toxicology models. And really this follows the example that I've shared with you today. It's about being able to develop a system that's human relevant with a high predictive value. Importantly, we believe in the value of using experimental designs set forth by third parties. And if those experimental designs are followed, we can then evaluate the model performance in terms of its reproducibility and its variance. And then really we're at the last, we think, or the last steps with respect to liver chip, not the last steps at all, but with respect to liver chips, it's about really educating users, and it's also about working with our colleagues in the regulatory agencies. And once we can work with these individuals and these groups and execute this path in its entirety, we believe that we can move forward with improved risk assessment. And if we get to that point, given that we're talking about 3 Rs today, we do believe that we're now entering, certainly for liver chip, a period of what we'd call coexistence. If you remember, we're proposing that liver chip would be placed in a workflow before one did an animal dose range finding study. Obviously, many more experiments need to be done, many more candidates need to be tested, many more donors need to be used. And of course, over time, we need to build more models. We don't just need to build liver models. The heart, for example, is another leading indication of drug attrition, the kidney, the gastrointestinal tract, and so on and so forth. Over time, the aspiration may be that liver chip or an organ on a chip might be able to sit nicely with one species rather than the need for 2 animal species. And at some point in the future, there may be the opportunity to do a full replacement. But you can see we've not put any time on our time arrow because they'll take a lot of hard work and a lot more data generation to get to that point. So just in summary then, to leave you with some concluding remarks, we would believe that the time is now for next generation toxicity assessment, and well-characterized human relevant models can show improved predictive validity, and that the model choice and experimental design are very critical to this success. We need to still work with our end users and regulators to understand their needs and what needs to be demonstrated from a data perspective to complete the path to adoption. But we're very aware that there's many questions that still remain, and we need to understand more about variability. We need to approach how we validate this technology. We need to think about standardization, numbers of donors to test, and so on and so forth. But what underlines it all really is that this is a collaborative community approach that with a defined framework we believe can help accelerate the adoption of human relevant models. And by doing so, we get to the ultimate goal here, which is to assure greater patient safety. And with that, I will say thank you. And I've realised my acknowledgement slide is not in this deck, but this, of course, would not be possible without the fantastic team we have at Emulate. The work that you've seen presented today took place last year in 21 actually now, sorry, yeah, it took place in 21. It was 16 weeks worth of work and involved nearly every scientist at Emulate. I've also been supported by external collaborators from AbbVie and J&J, as well as the scientific founder of Emulate, Professor John Ingber. So with that, I'll close, and if there's any time for questions, I'm very happy to take them.